Welcome everyone, and thanks for joining what is the third of our seminar series, sort of loosely on spiking neural networks. Our first speaker today is Alessandro. Alessandro and I actually first crossed paths in the first year of my PhD when he was a master's student in the lab, and we were both doing experimental work, which is pretty different from what we're both doing now. And yeah, I saw he had this paper out, and I think it will be really interesting for the audience here. So yeah, Alessandro can tell you the rest.
Okay. Yeah. So hi everyone. Thanks for joining, and thanks Marcus for organizing all this. I'm going to talk about some recent work that we've been doing in the lab, kind of building some simple models of the dentate gyrus and trying to see how different cell types interact and how they can help for doing pattern separation. Okay, so one situation you might be familiar with is trying to find your car in a parking garage. So this is something that really puts our spatial memory to the test, and hopefully you can find it in less than 6 months. But in order to do this, our brains really need to figure out how to tell apart very different or very similar environments and try to kind of disentangle their neural representations, so that even though everywhere kind of looks the same, we're still able to tell that if our car is here or if it's in the next slot or on the floor above or below. So how does the brain do this? Well, there's one region that's particularly associated with this function, and in brain slices, it really stands out because it has a very high density of cells. And they're packed very closely together, and so when we stain for a nuclear stain, we can see it. It's in very high contrast. And so what exactly this region is called the dentate gyrus. So what exactly does the dentate gyrus do, and why are there so many cells packed in there? So let me start by just showing you a bit of data about what we know about the dentate gyrus. So first of all, the activity of the principal cells in the dentate gyrus is incredibly sparse. So these are called granule cells, and generally only about 2% of the dentate granule cells are active in any given environment. And when they are active, they're very selective. They're only active in one environment, generally with only one place field, and they have kind of fairly low average firing rates. Outside of their field, they're completely silent. And so they're kind of very sparse and very selective in their firing. And the other kind of key feature of the dentate representation is the fact that very similar environments tend to have decorrelated population representation. And so this is what people refer to when they talk about pattern separation. So in the data I'm about to show you is kind of a very well-known experiment in which they take an environment and then they make many small changes until eventually the environment looks completely different than the first environment they started with. And what we see in general is that the dentate gyrus, the representation is very sensitive to small changes in the environment. So what we're seeing here, the points on the right represent two kind of very different environments. And so there we would expect that, the population representation is going to be also very different. On the other hand, when we have two very, very similar environments, now the dentate gyrus can still maintain some level of discrimination between these two environments and still kind of somewhat decorrelate the neural representation, much better than, for example, neurons in other parts of the hippocampus, like the CA3. Okay, and kind of one indication that this ability to separate similar environments is kind of behaviorally relevant is, here's one example that in old mice that have memory impairments, it seems to correlate with a loss of pattern separation and like a collapse of the representation, so that the same cell will fire even in very different environments. Like we see here, whereas on normal circumstances, both in dentate and in CA3, we would expect that if the environment is clearly different, then the representation would also be different. And generally, you don't have this, the same pattern of firing. Okay, so how does the dentate gyrus do this? How does it separate similar patterns and kind of disentangle their neural representations? And so to get a better understanding of this, why don't we take a closer look at the local microstructure within the dentate gyrus? So as I mentioned, the main excitatory cells are called granule cells. So they receive feed-forward input from entorhinal cortex, integrate it, and then send their axons forward to CA3, and then that kind of propagates forward throughout the hippocampus. Then they locally interact also with a variety of interneurons. So some interneurons provide feed-forward inhibition. There are other neurons that function as feedback. And here, the architecture is actually slightly unusual in that the feedback operates mainly through a set of excitatory local interneurons. They're called mossy cells. They're recurrently connected, and they provide kind of simultaneously feedback excitation to the granule cells, but they're also driving these feedback inhibitory interneurons, which are specifically dendrite targeting interneurons. Okay, so to kind of get a better understanding of how all these cells are interacting, what are their different contributions to the overall pattern separation, we're going to abstract a little bit away from the biology and consider a more simplified rate neuron model of the dentate gyrus. And sorry to disappoint the fans of spiking networks here. This is a conductance-based model. So we have kind of synaptic conductances, cell conductances, but it's not actually a spiking model. And what we're going to do is we're going to see how does the representation change as we add in different cell types. So we're going to start very simple. We're going with the kind of the simplest possible network, just the input from entorhinal cortex and the granule cells here represented by these output cells. And we have this characteristic of the fact that there are very many granule cells. So you have this expansion of the representation. And one characteristic of the input is that it tends to be relatively dense with overlapping activity patterns. You can think of like granule cells, grid cells that have many place fields in an environment, and they're often active across many different environments. So they have this kind of very dense representation. On the other hand, in the granule cells, we would expect to see a very sparse activity pattern, and preferably something that's relatively orthogonal. So something that has very different activity pattern across different inputs, different environments, and so on. So, okay, so now what we're going to do is we're going to add different cell types. And what I'm going to show you what happens as we kind of progressively take a kind of building blocks approach. Like Lego blocks, we add, what happens when we add feed-forward inhibitory component, a feedback component, some combination of these, or these excitatory mossy cell recurrent excitatory neurons. And one thing I should add is that we're really focusing here on like the architecture itself, like it's sort of kind of in isolation. What kind of prior does the architecture provide to this system that can maybe help it to try to disentangle the representations, but kind of independent from any specific learning rule. And so to try to kind of reduce as much as possible all the different free parameters, rather than actually learning the weights through backprop or Hebbian learning rules, we're just going to sample the weights randomly and maybe do a little bit of optimization, just trying to tune the overall strength of each projection, but without actually doing any fine scale learning. Okay, and so as for the inputs, what we're going to do is we're just going to try kind of all the possible combinations of these 7 input patterns. So each input unit, there's 7 input units in this little simple little model, and each one is going to be active for exactly half of the time. And so if we do all the possible combinations of these, this is what it looks like. And what we're then going to see is what happens in the output representation, which we're going to kind of quantify in terms of these 3 metrics. So we're going to look at the sparsity of the granule cells. So in other words, how many units are active in response to a given pattern. We'll look at the selectivity. So for any given output unit, how active is it? How selective is it for one particular pattern? And then the discriminability kind of across patterns, how orthogonal are they? How easy are they to separate, to distinguish? So even though they're technically different, we might have some patterns that are very similar, but ideally we would like them all to be as separate from each other as they can be. And so we might imagine that some sort of ideal intersection of all of these these metrics might look something like this. So where we have, for here, every row is a different output unit. And so we might have every unit is specifically selective for one pattern. There's only one unit active for that pattern, and they're all kind of trying to be different from each other to be perfectly orthogonal. Okay, this is the kind of the ideal scenario. So let's kind of see what happens as we play around with the architecture. And so starting with this kind of very simple input-output network, which is kind of going to be our baseline here. And so we're just have like a random expansion of the representation. And so we're not really expecting anything, any huge change in the ability to represent these patterns here. And so as we can see, the activity is kind of very distributed, unselective. And actually, we see one other challenge with kind of this particular set of input patterns that we've constructed is that you might have some combinations where, by chance, you might have multiple units that are active at the same time, and multiple of the inputs that are active at the same time, others where there are fewer inputs active at the same time. And so one of the challenges is also, how do you avoid having all the neurons become selective for the strongest input patterns? So here we see kind of a summary of these three metrics. The red line indicates like what the representation among these 7 input neurons looks like, and then the black line is the representation in the output cells. And so we see that there isn't a huge difference in any of the metrics. And for selectivity, it looks a little strange. That's just because for the input, every unit is active exactly half of the time. So there's kind of exactly half selectivity, whereas in the output units, you have some that are just firing completely unselectively all the time, and then others that are maybe have by chance they got slightly weaker weights. They are only selective for a much smaller number of the patterns. Okay, so what happens as we begin to add inhibition into this network? Well, with feed-forward inhibition, we now have at least a separate independent readout of this input. So at the very least, that can help us to try to normalize this level of excitation across all the different input patterns. And so I'm just going to show you as we kind of turn up the knob of the inhibitory strength. We can see this kind of slight flattening out of the representation across the different patterns. But this is still a very coarse control. As we start to really crank up the inhibition, we start to lose a bunch of the output units. They become completely silenced. On the other hand, we're not really getting any gains in selectivity it's the same strong units that are just active all the time. And so we're not really spreading it out among the output units. And if we try to just pick some optimal value for this inhibition, we get some improvement over just the baseline of without any interneurons, but not huge. So, and then let's look at what happens with feedback inhibition. Well, we actually have a similar problem. So if we had maybe a classic like winner-take-all system, you might think that, you would expect that this would work quite well. But one requirement for that to work well is that there has to be some structure in the input weights. So if a different unit receives the most amount of excitation for every given pattern, then a winner-take-all kind of lateral inhibition might help to clean up the noise and really sharpen that representation. But in the absence of that, what you have is that if some units happen to get slightly stronger weights than others, then those units are going to win all the time. And so now, if we crank up the inhibition, we get something kind of similar where, again, we lose out a bunch of the units and the others haven't really increased their selectivity all that much. And so we see still, a little bit of improvement over the baseline, but not huge. Okay. So something interesting happens if we reroute the feedback inhibition to go through these recurrent mossy cell-like excitatory neurons. And the main thing is that now we have, thanks mostly to these recurrent connections, we have this nonlinear amplification mechanism. And so even if you have very, very weak activity in the output cells, you have this mechanism that can really sort of exponentially ramp up the amount of inhibition and give you a much more dynamic range for it. And so what we have now is that we can really have different regimes of inhibition that can actually help to really make the population not just sparse, but actually selective without completely silencing any units and without just having the same units active all the time. And so we really get kind of quite a substantial kind of improvement over these three metrics that we've chosen. Of course, we're still not, still not the perfect orthogonal pattern, but we're still doing quite well with completely random connectivity. And just to show you kind of that actually these recurrent connections really are the crucial element here for getting this to work. And the reason is because if we remove them, then we don't have this sort of amplification mechanism. And so now we really have not really any benefit over what we had earlier with just the inhibitory interneurons. And so it's really like this ability to have this sort of amplification in the system that can really benefit and give you a much more flexibility in how you control inhibition. And then obviously this is what we can do with purely random connections. I'm sure learning, of course, is very important, and so let's see. So yeah, of course, that's kind of one of the future directions that I'm working on, is to try to see, well, of course, we would expect learning now to be able to refine these connections, especially if learning can help us get a slightly more structured input weight matrix. Now that gives the feedback inhibitory neurons a lot more material to work with, so it's much easier to get a winner take all system. And so maybe if you have the right architectural prior, now with just a little bit of inhibition of learning, you might be able to do quite well without putting too much effort into having something very complex. And so that's kind of the direction I'm pushing on, especially trying to see how it's actually known that there is some feedback connections from CA3 that go back to the dentate gyrus. So trying to see if that might have a role in guiding learning and the dentate gyrus. So anyway, that's me. I'd just like to thank everyone in the lab here for their help, and all our funders, and yeah, thank you all for listening. And if you want to check out play around with the model yourself, you're welcome to, or you're welcome to get in touch.
Cool. Thanks very much for the talk, Alessandro. It was really clear. Yeah, I mean, does anyone have any questions which they'd like to ask?
So I just want to clarify something, if I may. Thanks. A really nice talk, really nice figures also. It's really clear what's happening. So at the end, you say that you still have to implement learning, but it's not so clear to me. How do you set up the connections right now? Is it just kind of global inhibition and global excitation loops? Or is there some pattern that the random connectivity or?
Yeah, so we just choose, we have random distributions for each of these, there you go. So for each of these projections, we'll have a random distribution that we're sampling weights from.
I see.
And we chose, for the excitatory connections, we chose log normal distributions because they seem to be somewhat ubiquitous in a lot of these systems. And for the inhibitory connections, we just chose uniform distributions.
Great.
And then all we're doing is we did some optimization. We did a sort of pseudo evolutionary algorithm that optimizes for just like the parameters of this distribution that we're selecting from, because we to try to give it the best chance it can with sort of one hyperparameter per projection, basically.
Nice, thank you.
All right. I was thinking just longer term, you could imagine like a really big space of these of the sort of possible networks with the task, right? Have you had to think about ways in which you could explore like a much bigger space of models?
You mean like try to kind of expand the number of different like interacting populations, and for example, or?
Yeah, that sort of thing. I mean, I guess with additional layers or, yeah, I mean, you could imagine adding like feedback recurrent connections to like, all the populations you've shown here as well.
Right.
Yeah.
Yeah, I mean, I think, yeah, exactly. Like the number of possible connections, really start, and especially if you start adding additional populations, it really starts to explode. Like all the number of possible combinations. And then it does become a little tricky to try to really optimize all of the connections at once. And in fact, there's some models that I didn't show, but that really have even more connections. In some, at least for our case, like for example, when we add this feedback excitatory connection, it didn't make a huge difference in this particular little toy system. Although, yeah, so I think it kind of, yeah, it depends on what your objectives are. Like, yeah, we could easily scale it to have many more populations. It'd be hard, maybe a little harder to interpret what's happening. And so, yeah, so we try to at least stay a little bit grounded in whatever circuitry exists in the biology, and then hopefully see. And I think the more interesting question really is to now see how this interacts with learning rules, and I think in fact we suspect that this feedback connection might actually have something to do because there's a kind of a specific dendritic targeting to these feedback connections. We suspect that might actually have something to do with helping the mossy cells potentially drive learning at the feed forward synapses.
I have a follow-up question if there's time for that.
Yeah.
So, if you just consider only the feed forward rule weights, and I know that there's a couple of learning rules like the BCM rule that's explicitly really good at separating out patterns.
Yeah.
And create competition between different input patterns and things like that. Do you think that you could get like better performance in your metric of performance if you train the forward weights in such a way instead of especially in the selectivity and sparsity, for example, instead of using the random connectivity you're using now?
Yeah, I think, yeah, I would hope that you could definitely do a lot better with training the weights. I think one of the challenges, and I mean, maybe this might be slightly artificial by the way we set up the problem, but that you need to have a way to select the problem is that the patterns are so overlapping that you have some patterns where maybe all 7 of your input units are active at once, right? And so you need to have a way to be able to select for like the 1 hot pattern without having that, without also selecting for the 7 hot pattern. And so it's almost like you have to do some sort of like nonlinear separation of or something a little bit akin to an XOR type of problem, right?
Because they're not orthogonal inputs, basically.
If we're talking about the input space, like in the input space, so the output neurons have to be. So if we only had like this input output with a BCM rule, for example, here, then that might be a little bit of a challenge. Trying to say I want to select for the 1 hot pattern, but not the all hot pattern. Right, and so there, I think having that a little structure in the inputs can help, and then so maybe the BCM can help try to create that, but then the feedback inhibition can clean it up and to suppress the unwanted patterns. So yeah, but I think that's something that I'm kind of looking to explore to see like if we compare a few different of these like BCM or other Hebbian learning rules or even 3-factor learning rules, then what can we get? What representations do they converge onto?
Is there time for one more?
Yeah.
So
yeah, go ahead. Yeah.
I have sort of a similar question to Marcus's in terms of sort of like the combinatorial sort of like explosion of the different variations of cell types you have. So here you've been sort of like adding 1 cell type at a time, right? You started with like the feed forward inhibition, then you added the next one, then you added the next one. To what extent do you need like the previous, like you're adding cell types until you got to the discriminability.
Right.
To what extent do you need the previous cell types that you added? Like, can you just get away with like the excitatory feedback and without any of the like inhibitory feedback and the feed forward inhibition?
I think the feed forward inhibition you might be able to get away with removing. in this case, the way the network is set up is that actually you need some form of inhibition, and it just happens that in this case the inhibition is kind of taking the long way around via these excitatory neurons. But you can't, if you only had the excitatory neurons, now you have no source of inhibition. So for sure you need, like, the excitatory neurons on their own can't do anything without the inhibitory neurons. They're not going to really help. But yeah, we did try variations with only one or only the other, of like, yeah, the feed forward or the feedback, or so on. So I think this structure seems to be like the most useful one for this particular problem. Yeah.
And a thought that I was having on this in terms of like, how do you explore this large space of combinatorial different populations? I wonder if you can sort of like fix some sort of random weights and then optimize for discriminability, but only optimize how much each population can participate, and put in some regularization penalty on that, some sort of sparsity penalty. So like, what is the fewest number of populations that you need to get to the discriminability results? And then you can explore like whatever absurd number of architectures you want.
Right, I see. Like if you optimize across all of the different projections and including the number existence of one or the other population. What's the
and penalizing for sparsity.
And yeah, so we did something, in a sense, we did something not too dissimilar for that. So when I didn't really go into the optimization that we used, but what we're using is a multi-objective optimization algorithm that tries to kind of balance these three different metrics and get some sort of optimal performance across all three. And there is a little bit of a trade-off, because if you have really high inhibition, you might lose out on discriminability. You might lose out on selectivity, because you have like some neurons that are just not selective for anything, and then some neurons that are firing all the time and some that are never firing. But of course, you're going to do really well on sparsity. So we tried to kind of have something that is a little bit similar to an evolutionary algorithm that just kind of tries a whole space, not a full grid search, but a whole space of possible like strengths for each of these projections and for these hyperparameters, and then try to find some sort of balance of optimality for these three parameters. And then because here we don't have so many different, like we only have 3 different or 4 different populations, so we could basically try every combination, and kind of settle on the fact that the one that seems to perform best is when we have the system with these excitatory mossy cells, or this one with or without this feedback connection, but certainly with the recurrence. And so we didn't really have to include that as a separate parameter in the optimization. We could just run several different optimizations, and in each case just say, for this particular configuration, we'll optimize all the possible every possible combination of parameters and do kind of a pretty large search, and then see what is the best we can do for this configuration. But yeah, I think you could include you could include as a parameter of the optimization itself whether or not you have this or that a population. Of course, that's gonna you're gonna need a lot of compute to do that, because now you're really exploding the number of simulations you have to run, and so yeah, it's going to be very compute intensive.
Well, a lot less human intensive.
Yeah, right. Yeah.
It was cool talk. Thank you.
Okay.
Cool. Well, thanks very much, Alessandro. I really like the building blocks approach, and it's also nice you tried to draw from the biology rather than
right. Stay a little bit grounded and try to keep things as simple as we can, and try to see if we can get some understanding. So anyway,
I really like the approach.
And all right.